Today's our topic, that name is the bacterial infections by the aerosols. Means Mycobacterium tuberculosis. That is our new topic. That is the MTB. Right? Mycobacterium tuberculosis, which is transmitted by aerosols, means which is airborne transmission of the disease, which is transmitted by the droplets. Right? These are the transmitted by the aerosols and the droplets. Means there are three types of infection transmitted. First one, this is airborne. Second, foodborne. And third one, waterborne. Out of them we are talking about the airborne, because which is transmitted by the airborne. Let's talk about our next one. What is called as the Mycobacterium? First of all, there are 20,000 species. Now we are aware about the species and the genus name. There are 20,000 different species of the Mycobacterium which are observed. But out of them, only few species which is called as the infectious or the pathogenicity developed in our body by the Mycobacterium tuberculosis. Okay. So MTB, this is the acid fast bacilli group of the bacteria. Right? See, this is the MTB. We are talking about them. AFB group. This is the acid fast bacilli group of the infections. There are two type of the AF bacteria, non-tuberculosis and tubercle bacteria. Means ki AFB, two types. One is NTM, another one is the MTB. Non-tuberculosis bacterial and the MTB means ki Mycobacterium tuberculosis. This is another species which is not coming from to the mycobacterial group of the species. We are talking about the Mycobacterium tuberculosis in a detailed manner. Let's talk about them. MTB. Mycobacterium tuberculosis, this is the acid fast bacteria and their bacterial characteristic, which are bacterial characteristic which are, first one, mycolic acid which is present into the cell wall, gram negative bacilli. The third one, transmitted by the aerosol and droplets. This is the basic characteristic of the MTB, means ki Mycobacterium tuberculosis. In our body, into the human body, only two things were not infectious conditions which are appear, only that is hair and the nails. This except all the body's part which is infected with the MTB bacilli or the MTB bacterium. Right? Out of them there are many species which are observed into the MTB. First one, another one MTB means ki which is caused for the tuberculosis. M lepra cause the leprosy. M avium transmitted by the birds. Africanum. This is all the another species who causes related of the MTB group of the bacteria, right? Now the types of the MTB means ki there are types of the MTB. Well, how many types of the MTB bacterial which are infectious to our body? The generation or the double time of bacteria, this is 18 hours. It's too much. Generation time or the doubling time of the bacteria, which is the 18 hours. This is very long time to take for the generation, means ki doubling for the MTB bacteria. Okay. This special cell wall characteristics due to this bacteria stain with AFB stain, acid fast stain, which are required. Okay, the acid fast stain which is required because of this special cell wall characteristic, which is what? That is the mycolic acid, lipopolysaccharide materials which are absorbed into higher amount into the bacterial cell wall of the F bacteria. Let's talk about them. How to be identified the MTB bacteria. So obviously any bacteria which is identified by the three phases. First one is the staining, second one media inoculation, and third one is the biochemical test. These three things which are observed for the identification of the bacteria. Now first one, staining. Which stain which is applied? AFB. Let's talk about them stages of the AFB. See, this is the gram negative bacilli. So the bacteria would be appear in a reddish color and the background is the purple bluish. Bacteria which is appear into the red color and the background which is observed into the purple or the bluish color. Let's talk about the staining name. First one, this is the use for the cell wall stain. That name, a 1% carbon fuchsin. This is the cell wall stain name, 1% carbon fuchsin, which is used for the staining. Means ki onto the slide, a 5 ml, around 5 ml use of the carbon fuchsin added onto the slide. Cover the slide with the 1% CF. Second stage, warm up the slide and rest for it 10 minutes. What is the characteristic? Means ki this is the staining patterns, this carbon fuchsins and this warm up for slide. Means ki just give the pores will be going to be open due to of the harsh parametrical apply, because the lipopolysaccharides and the lipid content and the mucolic acid, means ki the higher lipid content which is not penetrated any other staining patterns into the cell wall characteristic. Due to of that reason, another parameters we have to apply like the heat. A small amount of the heat which is applied onto the cell wall characteristic, which is reason behind of the open the porous and penetrate this is CF into the cell wall characteristic, and after the 10 minutes it closed down all the porous and fitted down that CF into that mycolic acid characteristic of the cell wall. Then after fourth stage is apply 25% H2SO4. it gives the decolorization. Decolorization means excessive mycolic acid related of the carbon function which is not attached to the cell wall. Remaining of all these conditions will be going to be washed out, so that crystals of the carbon functions which are not hindered by during of the viewing of the slide. So the excessive amount of the carbon functions which are going to be removed by the use of the 25% of the H2SO4. Wash with the water. Sixth one, apply counter stain. What is the role of the counter stain? Counter stains we just give the color contrast between the bacteria and the background. So we can identify it a very easily manner what is the type of the bacteria with the background. Okay. So counter stain means ki 0.1% methylene blue which is applied onto the slide and rest it for the 30 seconds only. This is the second. This is not a minute. Just apply for the 30 seconds only. And under the oil immersion lens, bacterial slide of the fluid we have to observe. The bacteria would be always observed in a round manner due to of the lens. You know the lens curvature which is the round, so we have to measure into the round. The bacilli which is always appear into the chain or the pattern like are always observed into the reddish color. This is the patterns of the Chinese letter of the conditions, and this is bacilli which is appear, and this background which is observed by as according to the methylene blue applied, it is observed into the purple bluish color. So the bacteria would be going to be appear as the round color, as the red color, and the background which is in a blue color. Right. Next one. Second phase is the media inoculation. Here a special media means enrichment media which is you applied to cultivation of this bacteria. That name is the algae media. Algae media. Algae media means ki Lawrence Jensen media. In this media, egg metallic color, greenish metallic sheen color type of the media which are appear. The extra ingredients, extra richness type of the ingredients which is applied, which is the egg to making down of this algae media. Next one. After the cultivation onto the media, this is the rough, buff and the tough colony, rough, buff and the tough colony which is observed onto the algae media. Right. See, this is the conditions we are to applied onto the agar rose media. Enrichment media, that is the algae media, special media which is applied for the cultivation of the MTB bacteria. Okay. Next one. Stages of MTB. The first one, primary means ki first line, and the second one is the second line. Third one, HIV co-TB. Fourth one, pediatric. Fifth one, relapsation. These are the types and the stages of the Mycobacterium tuberculosis. This is the types, these are the stage. In a first line, this is the two drugs which are registered for the MTB bacteria, that is a first line which is rifampicin and isoniazid. These two drugs, if the patients which is registered by during of the DOTS process, means ki given by the medicine and they are not completely recovered by this first line drug, it is indicated that the follow up we are taken down of this taking drugs if we are not giving the less of the bacterial load, which is called as the resistant of the first line factor, and this is why rifampicin and the isoniazid factor which are appear, and this is called as the first line condition, means ki this is called as the MDR tuberculosis. This is called as the MDR tuberculosis. Okay, what is called as the MDR? This is called as the multi-drug resistant tuberculosis. This is called as the first line tuberculosis. Okay, these two drugs which are applied onto the patient and it is not affected by the patient's bacterial load. So this is caused by the MDR tuberculosis. The next one, second one, that is the XDR TB, which XDR means ki extremely drug resistance of TB. This is the kanamycin or capreomycin. This is the injectable drug which are used. If this type of the drugs are not using these drugs which are not applied, means bacterial load which is all remaining, which are not affected factor on killing the bacterial, this is called as the XDR tuberculosis, right? This is called as the XDR tuberculosis, right? Now another one, due to of the immunity, means ki if the patient is suffer with HIV who caused the TB, why? Due to of the suffering with the HIV, because in these conditions the HIV infected patients whose immunity system is very less, due to of that things, this is causes for the tuberculosis factor. Next one, pediatric TB, means congenital, in a congenital way, means which is transmitted by mother to fetus, mother to the fetus. This is fetus or the baby. This is called as the pediatric TB. Next one, relapsation TB. Relapsations means again and again causes for the tuberculosis. This is called as the relapsation. What is called as the relaptator factor? One time TB which is caused, it will be cured. After some time, the infection is again observed. This is called as the relapsation. And relapsation type of the tuberculosis, the treatment part which is different. This is called as the conditions of the observe. This is the relapsation factor. Next one, and infectious factor, again relaptator factor, In these conditions, whatever treatment we were given during of the first time exposure of tuberculosis, after the relapsation factor, we can't observe as the same dose given as the first time previously causes the disease conditions. Okay. Next one, this is the relapsation factor, and another the test which are primary and secondary. BCG, this is the vaccine given at the birth time. Right, the BCG vaccine which is given to the birth time, which is gives the primary immunity to the patient. Okay, so this is the causes for the disorders of these conditions. This is intestinal tuberculosis. This is the respiratory related of the tuberculosis. In these conditions, what type of the specimen we have to require? Like, first of all, CSF. This is the two types. Let's talk about them. This is the two types of the tuberculosis which is appear, that is pulmonary, and another one is the extra pulmonary. Pulmonary and the extra pulmonary. Pulmonary, which is related by the cough materials, and this is it related which is not related to the blood, not related to the blood. This is called as the pulmonary tuberculosis, and this is related to the CSF, blood, tissue, pleural fluid accumulated into the pleural cavity portions due to of the Mycobacterium tuberculosis infection. This is the cerebrospinal fluid, this is the blood, this is tissue, pleural fluid, and serum, plasma. These are using for the extra pulmonary conditions of the factor, extra pulmonary factor of the disease. There are two type of the samples we have to taken in identification of the tuberculosis clinical approach. This is pulmonary and second one is the extra pulmonary. Right. Next one, treatment doses patterns by the dots. How to be caused for the treatment? The treatment, direct observation short course chemotherapy. This is the full form of the dots, which is the given as the dots factor. HIV patients gives the two types of the condition, HIV plus tuberculosis factor of the treatments which are given by the ART center. In this condition is according to the following: a follow-up by every month. Means whatever treatment you are started by to the patient, it always gives the follow-up in every month. So we can identify by the staining patterns and the media identification factor, media inoculation factor, what the type of the growthal patterns which are appear as the higher level or the lower level. If we are given to as a treatment in a first line factor, so it is indicated that the growthal patterns for the every month it is going to be inhibited, not for the growthal patterns which is going to be higher. If it is going to be inhibited, it is indicated that the bacterial treatment, whatever doses treatment we are taken to them of the patients, which is going to be sensitive. If it is not given to the sensitive, which is called as the resistance. So we have to change our regimens. Okay, regimens means ki given the pattern doses, we have to change our regimens. Right. Next one, direct observation short course the chemotherapy. Another, this is the RNTCP program, which is run by the tuberculosis factor of the disease, means ki tuberculosis conditions. These are the organized by, means ki this is treatment under by the RNTCP control, means ki revised national tuberculosis program. Due to of these conditions, there are observed factor. RNTCP program full on work with the tuberculosis factor. Okay.